This method provides an alternative to conventional fluorescence labeling and flow cytometry analysis procedure, which are time-consuming, costly, and incur the risk of altering the cellular function of the samples. The main advantage of this technique is that three-dimensional refractive index tomography and machine learning are label-free and quantitative methods that enable a rapid and accurate lymphocyte identification.
The implications of this technique extend toward the therapy of blood cancers and autoimmune diseases, as identification of lymphocytes can be crucial for disease diagnosis and appropriate treatment application.
Though this method can provide insight into lymphocyte populations, it can also be applied to the analysis of other single cells of interest, including bacteria.
Visual demonstration of 3D quantitative-based imaging technology is critical, as it facilitates a clear instruction of how to perform the technique and can provide insight into its applications.
Begin by collecting each lymphocyte subset by fluorescence-activated cell sorting. For optimal imaging, dilute each cell sample to a concentration of 180 cells per microliter of RPMI medium, and slowly inject 120 microliters of the first diluted sample into an imaging chamber. After confirming a lack of bubbles within the chamber, place a drop of distilled water onto the objective lens of a 3D quantitative phase microscope and place the imaging chamber onto the translation stage of the microscope. Adjust the stage so that the sample aligns with the